My name is Myron Suchak. I'm a professor of immunology at Queen's University in Kingston, Ontario, Canada for the past 31 years. I'm also the lead author of this review article together with Samar Abdulkahelik and Michael Herniak. For the past 5 years, we have been working on the role of glycosylation receptor activation with particular focus on receptor tyrosine kinases such as nerve growth factor TRK receptors, the epidermal growth factor receptor EGFR, the insulin receptors, as well as the specialized pathogen sensing receptors called the toll-like receptors. We have discovered that there is a mammalian neuraminidase 1, also called New1 sialidase, and matrix metalloprotease 9 MMP9 crosstalk on the cell surface, which was found to be essential in regulating ligand activation of glycosylated receptors. Within this crosstalk receptor signaling platform, we found that ligand binding receptor induces G protein coupled receptor signaling by G alpha proteins and MMP9 activation to induce New1 sialidase, all of which form a tripartite complex with a receptor at the ectodomain on the cell surface. This novel signaling platform is depicted in Figure 1 in the review article. To visualize the signaling process, we produced and recorded an animation video for a visual impact of this novel receptor signaling platform in regulating glycosylated receptor activation.
On the cell surface membrane, we have identified a novel signaling platform that is critical for the activation of the glycosylated tyrosine kinase receptors like the nerve growth factor TRK. This signaling platform consists of a tripartite alliance made up of mammalian neuraminidase 1, also known as New1 sialidase, matrix metalloproteinase 9, also known as MMP9, and G protein coupled receptors. New1, which is part of the multi-enzymatic complex of beta-galactoside, protective protein cathepsin A, and the elastin-binding protein, forms a complex with a receptor at the ectodomain with MMP9 and the G protein coupled receptor. The ligand binds to the receptor, which initiates a conformational change to induce the GPCR signaling process tethered to MMP9 via G-alpha-1 proteins. Activated G proteins subsequently activate MMP9. It is the elastin-degrading activity of MMP9 that fits well within this novel signaling paradigm of tyrosine kinase receptor activation. Direct removal of the elastin-binding protein from the multi-enzymatic New1 sialidase complex by activated MMP9 is proposed to induce New1. Activated New1 requires protective protein cathepsin A to perform its enzymatic activity. Activated New1, already bound to the ectodomain of the receptor, specifically removes the alpha-2,3-Cl residues of the receptor to facilitate dimerization. This receptor dimerization is essential for tyrosine kinase receptor phosphorylation and subsequent cellular signaling and activation.
This receptor signaling paradigm, as depicted in the animation video, predicts that glycosylated receptors are in alliance with a functional GPCR signaling complex. Our data showed that ligand binding to respective receptor induces New1 sialidase within a minute, and that this activity is completely blocked by G-alpha-I sensitive pertussis toxin. The rapidity of the ligand-induced New1 sialidase activity mediated by the ligand-bound receptor suggests that glycosylated receptors like TRKs, EGFRs, insulin receptors, and toll-like receptors form a functional signaling complex with G-alpha proteins and GPCRs. If this hypothesis is correct, we should be able to activate New1 sialidase activity with GPCR agonists. We have reported that GPCR agonists like bombesin, LPA, cholesterol, and angiotensin 1 and 2 induce sialidase activity within a minute, and this activity was blocked by G-alpha sensitive pertussis toxin and MMP9 inhibitors galardin, piperazine, MMP9 specific inhibitor, and silencing interference of MMP9 messenger RNA. It is noteworthy that the GPCR agonist can induce sialidase activity in the live macrophage cell assay, but the GPCR-mediated signaling events occurred when New1 and a functional GPCR are tethered to the ectodomain of LPS toll-like receptor 4. Similar findings were observed using receptor tyrosine kinase RTK expressing cells. These results suggest that GPCR agonist activates sialadase activity only when U1 in a functional GPCR are tethered to a glycosylate receptor. In the review article, we also attempted to translate this novel GPCR signaling platform to human disease. There is a very extensive literature on GPCRs, including those that have dysregulation of function in association with a growing number of human diseases. The concept of GPCR dysregulation would include two mutational forms of GPCRs. Firstly, the receptor loses the ability to bind agonist or signal called the loss of function mutation, or the receptor is in an active state in the absence of agonist, the gain of function mutation. Indeed, there are a number of constitutively active GPCRs and a large number of mutant GPCRs as a cause of human disease. To date, over 600 inactivating, almost 100 activating mutations in GPCRs have been identified, all of which have been shown to be responsible for more than 30 different human diseases. According to the novel GPCR signaling platform as illustrated in the animation video, we are proposing that there is a common master GPCR tethered to these glycosylate receptors, to which many different GPCR receptors can heterodimerize. Targeting the molecular GPCR signaling platform tethered to RTKs or TLR receptors may provide important pioneering approaches to disease intervention strategies. Moreover, there are multiple orphan GPCRs for which the natural agonist is widely unknown. Given the large number of human GPCRs, estimated to be more than 800, additional examples of pathogenic mechanisms in drug therapies are likely to be discovered in the future. Indeed, 50% of human disease-related drugs in the market target GPCRs, and 25% of the top-selling drugs to treat hypertension, migraines, and stomach ulcers have been designed to target GPCRs. The future research focus might be ultimately directed at macrophage-mediated inflammation. It is well known that macrophage-mediated inflammation has been associated with many human diseases. I thank you for your attention and interest in this review article.